Okay, so maybe take this from Dr. Ngọc as the guideline. So I can paste here. Can you see that? Everyone see that? So normally, yeah, your friend did right. Chung did it correctly. So normally we have the sequence of DNA as double strand, and we have the positive strand from the 5 prime to 2 prime like this. And as a consequence, when we have uh, the positive strand this way, we have to read the negative strand from the reverse way. Okay, so from 3 prime to 5 prime. But when we see alone the negative strand in this way. Okay, so here you given the negative strand alone. So the first step you have to do is just that you have to reverse this. Okay, so to reverse this, so for example, maybe I remove everything. And, let me remove first. And so, for example, from this, I have the 3 prime T. The 3 prime will take the T here, okay, and C, and so on, T, etc. And we have the last 3 T, and peptide prime here. Okay, and afterward we have the thing that Chung did wrote. Maybe I press it as the form of text box. Maybe easier to follow. Okay, and I make it bigger. Okay, so as you might see here, so after you write the DNA sequence, okay, you write DNA sequence in the correct order, 2 prime to 5 prime of the negative strand. You write the complement nucleotide as your friend mentioned. So the T unit complement T A, G C, G C here, and 4A with 4T here. Okay, so on. You will have the complement DNA sequence or the positive strand. Okay, so what's next, Chung? What will you do next to?
So for the mRNA,
Okay,
uh, the sequence would, I think it would be the same as positive strands, but replace uh, T with U, because uh, the mRNA also is complementary from the negative strands, but it has U instead of T.
okay. So what we will do is that we copy this and we replace all of the T by the U, right?
Yes.
Like this, like this, and like this. Okay, so we will have this one. So basically, the mRNA is the positive strand that you replace on the T and the U. So what is the last step?
For
Peptide
the
sequences.
peptide, the first one, it would be a UG, since it's the starting codon.
Okay,
And
and can you point out where is AUG?
uh,
Use
mm,
the pen to highlight, to
okay.
circle.
Uh, uh, Sorry. Okay. Here.
Okay, so that's just the methionine, right? Or start
Yes.
codon. And what do you do next? You have the sequence of the peptide.
And each of the three, uh, each of three
Nucleotide.
will
Okay, except the three nucleotide.
code for a acetylene.
Okay, acetylene. And so what you have to do, thanks to what you have to do after you locate the start codon here, a UG, is that you keep the block of three, or you keep the frame. So each frame will contain three, or we call that a codon, and you have a UG here. So you keep going. You have to keep the block of three, keep reading the block of three. Okay, and here we have. After we have that, we can look at the bottom of the exercise sheet, right? Dr. Ngọc gave you the very nice table. Maybe I can put it up here for you. Okay, and we have this table. Okay, so after you have the codon, you will look into the table and to say which peptide or amino acid you have. So for example, you have here AUG. What you have is AUG is methionine here, and you have M. Okay, next one CAU. You look here, C here, A here, and U here, histidine. Okay, and to know the abbreviation, we also have the table down here, and we have, let me press here, table. Okay, so we have two tables, one for codon and one for abbreviation. So here we have CAU, we have histidine. Histidine will be H, right? And H, and UGG you can look here, and UGG tryptophan, and tryptophan, we have W, okay, and so on. You do that until the end, or when you have to, when you meet the stop codon. Oh, okay. So in biology, we have a thing called the central dogma when you have DNA, and after that you have mRNA, and you have protein. Okay, and this is called transcription. Okay, and here translation. Okay, so in here the process that you make the peptide sequence will be translation, and converting from the DNA to mRNA will be transcription. Okay, and here maybe I show you the solution for this. If you want to take a picture or something, yeah, solutions. Yeah. You have positive strand, you have the RNA, and you have the protein. You can write in 3-letter abbreviation or 1 letter, as you prefer. Okay, thanks, Jung. Very well done. So that's for the first one. And the second one, we have the second one here. So someone volunteer to do this? Please raise your hand. You have bonus point, to do this kind of stuff. No one volunteer? Không có ai muốn điểm à? Cái này lại gì? Okay, so since no one raised hand, Chung, you want to do it again?
Yes, I think I can.
Mm-hmm.
So first, I will write the mRNA of all the sequence.
Okay.
It sorry.
So how can you write that? So the intron will be in underline. What do you do with the intron?
I can eliminate it since it don't have the coding for the peptide.
Okay. So intron doesn't play a role in coding here, so we remove that, and we like this. We assemble these two together, two exons together, and after that, how can you convert that into mRNA?
So use the same as DNA, but so it will be T A with U. T with A,
Mm-hmm.
a G with C, and C with G. And I will and
So what you mean that you convert the A into G
A
and this?
into U, A into U.
Okay, okay. So why did you do the complement step here? Why did you do that? In
Because
mm-hmm.
I think it's code for
No, in fact, from here you have here what you have here is that you have exon and exon and intron here. That mean that you have from positive strand. Okay,
yes,
when you have from positive strand, that means that from the RNA you don't have to do anything. You just paste the whole thing here and replace the T with the U. Okay,
yes,
understood? Okay, so that's why your sequence of mRNA here will be this. Okay, if I make it smaller maybe. Okay, okay, if I make it smaller like this, you can see that A still A here. All both T will be UU, so on A. Yeah. Okay, maybe erase all this so you can easily see. So what you have to do with the exon is just that you replace. Yeah. uh, Okay, maybe so all right. Okay, you see that A still keep A, T become UU, A, C, 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 A, U, like that. Okay, and this part actually go up here because in translation the intron doesn't play a role here, doesn't have coding sequence. So in fact the mRNA is just this sequence. Okay, and the next thing is that to determine the peptide sequence. And as in the previous exercise, like your friend mentioned, we cannot just go from the beginning, just cannot go from the three first nucleotide. No, because we don't have the start codon. We need the start codon to begin the translation process. So again, the table come up. Okay, and we can see here you have to find the start codon, and the start codon begin by AUG or methionine here. And you can easily see that AUU is not the start codon. So what you have to do is that you keep searching from this direction until you meet AUG, and here you have AUG here. Okay, and that's the point where you have the translation begin, and that's why you have the translation. Yes. Let me paste it. Okay, amino acid sequence. And when you have the start codon, like previously, keep the frame 3 by 3. Okay, and here. Okay, and we have the translation, as you can see here. Okay, so this one, methionine, and CUC, uh, you can look C here and U here, CUC leucine, and you have L here. Okay, UCU, you go to U here and go UCC, sorry, and go C and go C, you have serine and you have serine here. Okay, you can also use the table on the right, the circle thing on the right to see. And for example, I want GUC here. So I go from G to U and to C here, and I have valine here. And similarly, CCA, C, C, and A, proline, so on. And you will have the peptide sequence for the whole thing. Okay? Do you understand that? Sorry. Do you understand that? Mm, okay, okay, Tung. Okay, that's it for the second one. Third one. The following sequence is located. Okay, Hải Anh lại anh.
We in here we will have eight codon is CTC, DCC, GTC, TCA,
Okay.
ACT.
So here what you mean is that we have one.
Yeah.
So we have the frame like this, right?
Yeah.
Okay, that's good. Do
And
you have any other possibility? So what is the different things between the one previously? So for example, we have here and this one. The thing is that do we have any information about start codon or not? No, right? Because we have in the middle. Okay, in the middle. So normally in the middle, so you can assume the start codon is somewhere in front of that. So there will be three scenario normally. Thank you, Hải Anh. You did it correctly with the first one. So the first one here, assume that we have the codon, start codon, and you have the frame here, beginning at the first three, and you keep the frame, so on, the three block. Okay, that's the first case. The second case, okay, I copy here. When we have the codon here, not the other two, the other place, so we have a translation here. Okay, that's the second case. The third case we have is here. We have the start here, not the start, the frame here, but the start codon is somewhere in front. Okay, did you understand? Because it's the combination of three nucleotides. So if you don't have any information about the start codon and you know that it's somewhere in front because this one in the middle. So there will be three possibilities. So this possibility always you can begin at the first. After that, you begin at the second nucleotide and the third one. Okay. And one thing that I want you to know about bacterial genome is similar to virus genome is that it doesn't have any intron. So everything in the genome of the bacteria will be processed to mRNA, will be translated if you have the correct start codon. Okay, so there will be no splicing. So normally you have exon, intron, exon, for example, from eukaryote genome, you have like that. But with bacteria, we don't have intron. Everything is considered exon. Okay, and so because this one we don't have information about the frame, so three possibility here, and you again use the table, yeah, and to know which amino acid you have. Okay, understood? Okay. And the fourth one, write the three possible correct codes for the polypeptide. Someone, các bạn nào làm không? Bạn nào làm được không? Các em có hiểu không đấy? Nếu mà không hiểu thì bảo thầy để thầy giảng một tí tiền. Mục tiêu là để các em hiểu còn làm bài thi. Có ai làm bài 4 hay không? Có ai volunteer làm bài 4 hay không? Okay, no one, so I can show you how to do this. Okay, so in this exercise is actually the reverse of the other three. So the other three, you are given DNA. After that, you go to mRNA and you go to protein. But in the fourth exercise, we are given protein sequence, and we have to write down the code for the encoding the protein sequence. So what we have to do here is just that we use the table that we had before. We use the table. Let me paste the table again. Yeah. Okay. So what we always go into proteins. We always goes with the table. And what we do with the table here is that we look at the table and we see which amino acids we have and which code we can have to translate into that amino acid. So, for example, we have leucemia, and you look at the table, there are 6 possibilities for leucine, and you can use any one of that. And so, to make leucine, to translate into leucine, we must have, for example, I can put UUA here. Okay, that's the first for leucine. And for the second one, you do similarly for valine. Look for valine here. For possibility of valine, you can use any of that. Let me go to this one. Valine, you have GUU. Okay, with the valine, cysteine. Similarly, we have cis here. This up there, so we have here we can use either one. Use U, for example, and lysine. Lysine we have here. So we have AAA, for example, and finally with the stop you can use either of the three here. So in this case, I use UAA. Okay. Okay. So you see that from the RNA here. This one is the mRNA, and we can have here the mRNA sequence to have the proteins will be. And if you are asked to write the DNA, you can also convert from an mRNA to DNA by using this one and here and like that. Okay, so the mRNA is here, and you with the DNA you just have to replace all the U by the T, PT, PT, T, T, and here we have T. Okay, if Dr. Ngọc asks about DNA, you can convert the mRNA into DNA like this. Okay, so that is the first possibility. So the second one, you just do similarly, and what you have to do here is just that you have to choose other combination to have, for example, leucine here. So instead of UUA, here I use UUG here. So we also have UUG leucine here. Next one, valine. Valine, instead of GUU, I use UC, for example, here. And cysteine, similarly, cysteine, I replace this by the C. And this imagine we have the G. And stop codon, you can use the UG, for example. That's the second possibility. Okay, so for the third, you choose other combination as well. So for example, here, as long as it's you have few different points, you're okay. So CUG, for example, and with the valine, we use the other A here. Cysteine, we don't have many choice, only have two, so keep it like that. And here you can use A or AG, and finally with the opcodon, you can use A here. Okay, so that is for the third. And you do similarly for this one. So anyone wants to volunteer for the B, part B here? I did this for you. So in the part B, you only need to do similarly. No? Okay, Chung.
So okay, for the his I can choose CIU.
The U will be his. Okay.
Methane is only a UG.
UG only. Okay, good. Serine.
Seri is UCU.
UCU. Okay.
Um,
Continue. Threonine.
Wait. ACU.
ACU. Okay. And
Maybe
similarly,
UIA.
UA, for example.
U.
Okay, that is the first one. How about the second one? Only two is, So how about the second one?
So for the second one, I can choose CIC for the first one.
CAC.
I
Okay.
can't change the second one. For the third one, it would be UCC.
UCC. Also works, right? Next.
Yes. For the next, mm, ACC
ICC works as well. And this
and
one,
UIG.
UAG, for example. Okay, so you got the idea, right? Let me mark your name. Thế Trung. Thế Trung. Okay, okay. Thanks, Trung. Okay, so far we know that the only thing that you can have to do in the genetic course normally is just that you do the DNA, you do splicing, cutting the intron, next convert to the mRNA and use the table. Dr. Now will not ask you to remember table, so just use that learn how to know how to use that one. Okay. So for the fifth one, I think this one is the most complex one. Anyone wants to try? Okay, now again, so I can show you. Okay, so transcription start. So transcription will take into account all of the thing here, except the promoter. So promoter doesn't play a role in the transcription. So transcription start, and we start here at this point. Okay, at that point start, and we have the precursor pre-RNA. In fact, when you have the pre-RNAs, that means you have all the exon and intron. So intron 1, H2 plus in2 and plus X3. Okay, so pre-mRNA, we have everything except the promoter. Okay, so with the mature mRNA, with the mature one, that means that you have to exclude the intron. Okay, don't count the intron in. So the mature will be the own exon. Okay, and translation start, and similarly like before, if we take into account where the translation start, we have to find the start codon. Right, let me erase this, and we begin by the order of the exon. So we begin with the mRNA, and the exon 1 will be the first one, next and next one. And so the first thing that we have to do is to find the translation start. We know that we have AUG is the RNA coding for the methionine start codon. So with the DNA, we know it's ATG. Okay, and so we look into the exon 1 and to see the first ATG that we can locate. So for example, you look at the A. You have the A here, A, no, ACT not, ATG here. Okay, so here we have start codon. Okay, since you have the start codon, you keep the frame. So 3 by 3. We have here. Okay. And so on. Okay. So here Dr. Ngọc asked you to write the first 10 residue. So what we have to do is also go to this thing and use the table again. Always use the table. Okay, so you have ATG here, so of course M AAT. You going to A and T will be U here. Okay, and we also have the second table Dr. Ngọc gave you. It's the one for the name, the abbreviation of the amino acid. Erase this. We have the name. Okay, so from here you have the we have the AAT and A. Look at A here and A here. So AAC, ASN asparagine, and we have N here. Okay. Next one, CGC. We have C here. Go for the G and go for the G. We have arginine here. Okay, and so on. You do that for the next 7. So in total you have 10. Okay, and I can give you the answer for this. So right here. This is the answer. So of course, like I mentioned before, start at the beginning with someone. For the A, here we have pre and the mature mRNA. And here we have the, like I mentioned, M and R and so on. We have, as you can see, the sequence here. Okay, and the translation stop when you have the stop codon. So normally you have to keep going. So here you have the frame here. Okay, so it's in frame. We have no stop codon so far. Stop will be UAA, UAG, or UGA, or in the DNA you convert that into T. So far we haven't had that. Next we move to exon 2 and we have the frame like this. BCA, BBC, okay, this, ATG, ACA, TGC, and finally we have TGA here. TGA here is the stop because we have the UAG here is the stop. So convert into DNA we have TGA. Okay, that clear? Okay, I assume now. So the sixth exercise. Can anyone do this? The sixth one. Okay, so no one answer. So okay, I think we have someone volunteer. No? Okay, Chung again. Yes.
So for the first part, I think that after each mitosis, the parent cell will divide into two daughter cells each
Okay.
time. So after 5 times, it will have 2 to the power 5
To the power
cells.
5 and equal 32. Okay, good.
Yes.
So about the number of chromosome.
So after the mitosis, each cell will have the same chromosomes as the parent cell. So each will have 24.
Okay.
Then uh,
Multiply by
enough by 2 to the power 5.
minus 1.
Yes. Yes.
And it's 24. Okay. So that's it. That's the whole 6 exercise. You just need to remember the formula. So here you have 2n multiplied by the number of cells minus 1, and you have that. Okay, I think there's no problem with this one. So the final one here, I guess no one gonna answer again, so I give you the the solution for this. So name of the sugar. From DNA, we have DNA, that means deoxyribose nucleic acid. So of course, yeah, we have put it in here, deoxyribose. Okay, yeah, with both the mRNA and tRNA, because they are RNAs. So we have the sugar here is ribose. Okay, here we also have ribose as well. And number of base present, name the bases present in nucleotide. And of course, with the DNA, we have A, G, C, T. Okay, and for mRNA, because we know that is RNA, so we have the T compared to U, and tRNA also similarly. Okay, so, and the function of DNA will be storage genetic info. Okay, this one DNA is storage of genetic info, and the mRNA. mRNA will play a role in the instruction of synthesized proteins. So instruction to synthesis protein, right? So with a protein synthesis, we must have the mRNA, and this will used to transfer amino acid to the mRNA for the synthesis. And this one arrives storage genetic info, and about the location. In this one, normally you can find a lot in nucleus, of course. Nucleus. And the other RNAs, we have cytosol. This one also cytosol as well. Okay. I think it's the first exercise. It's very short, kind of easy if you can follow. Okay, I think that's it. That's all for the exercise. And maybe now I want to call for attention for your bonus point for attending the class. Okay. Okay. So first, do you have any question? Any question relating to the exercise 7 exercise? If no, we can proceed to the attendance check. Okay. All right. Okay, I put it here.